All right, so my plan today is to give you an idea of the kinds of things you can do at the Indiana Center for Biological Microscopy. We're at IU School of Medicine in Indianapolis. We've been here since 1996. And I think rather than talking about everything that you can do here, given the fact that a lot of you have access to microscopes in your own lab or through your own core facilities or whatever, mostly what I want to do is emphasize what things I think are unique that we have here. So what I'm going to do first is I'm going to just briefly talk about the facilities and services we offer. And I'm going to talk about a couple of cool imaging systems that you may or may not be aware of that I think have a lot of legs but aren't necessarily appreciated as much as they should be. And then I'm going to talk about some advanced imaging opportunities that I think are unique to our core facility in Indiana at any rate. And then I want to talk about things we're planning on doing in the future. One of them is likely to happen. Both of them are likely to happen. One's more likely than the other, but we'll get to that. So we were established in 1996, and we've since that time become a core resource for 4 different NIDDK centers. We're a CTSI accredited core. We're used by more than one. At some point, you stop counting. We're used by more than 100 labs, way more than 500 publications. And I think the important thing about the facility is we're supported by 3 full-time PhD scientists who are trained in microscopy, digital image analysis, and can provide technical support on the design, procedures, analysis, train people, do studies for them, whatever. I'm the director of the center. Gosia Kamoska, she's Polish, her name is Malgorzata, but everybody calls her Gosia. She's the facility director. She runs the day-to-day procedures, and Connor Gilbronson was hired, I think now, something like 2 or 3 years ago, and he's an assistant scientist, and they each have their specific specialties within the center. I'm not going to go through this slide. I assume it's recorded so you can come back and look at it later, but the point is we've got, what do we got? We got 5 confocal microscopes. 3 of them also support two-photon microscopy. They all have their various strengths. And I'll be talking about each of these things in turn. Probably the most important thing here is that one of the confocal microscopes support resonance scanning, which is a very fast technique. You can do 30 frames per second. The other one is, and we recently got from the NIH, is equipped with 2 IR lasers for intravital microscopy, two-photon microscopy using 2 different IR lasers, has very special external spectral detectors. It also supports FLIM, which is something I'm going to talk about, Fluorescence Lifetime Imaging. And then we also recently acquired an ISS ALBA system that also supports FLIM, Fluorescence Lifetime Imaging, and it's supported with a white light laser, which is a laser, for those of you who don't know, is a broad spectrum laser. You can choose whatever wavelength you want to use. And then finally, we have this spinning disc confocal microscope, which is one of my favorite microscopes. We have additional systems. We got 2 epifluorescence wide field imaging systems for conducting assays of living, these kind of canned recipes that you get either from cans or molecular devices. I wasn't aware of the fact that that was going to be so popular, but a lot of people would like to do assays on multi-well plates, high content imaging. We also have now a Koia Codex imaging system. I'm going to spend a lot of time talking about that. Basically, it's a robotic fluidics operating system. And then we have 4 image processing workstations, which are equipped with ImageJ, Metamorph, Halo, which is really nice software, and Image Imaris 9.7, which also is very, very powerful software, as well as the manufacturer software. So with respect to underappreciated cool imaging systems, the first thing I want to bring out is this new tech is a system called the spinning disc confocal microscope. So probably everybody here, I guess, is familiar with laser scanning confocal microscopy. They probably have access to it, and they've probably done it. This is a thing that everybody comes to the center wants to do because they need an image for their grant or whatever. It's a very great technique. It has high sensitivity. It gives you high resolution. It does optical sectioning, so you do 3D volumes. It supports imaging of multiple probes. Why would you want to use a different technology? Why spinning disc confocal microscopy? Well, this has to do with the shortcoming of laser scanning, point scanning confocal microscopy. It's a kind of a silly way of collecting an image, but because you're using this pinhole to reject out of focus fluorescence, that means you have to actually scan a laser over the sample and simultaneously scan a pinhole over the imaged area, which allows you to collect an image of a spot, and then you scan that spot over the sample. That means necessarily you're going to go slow. You're not going to be able to collect images very, very rapidly. And the other thing is that since in order to collect images in a reasonable period of time, you're going to have very, very high laser flux, because you're going to have to collect whatever fluorescence you can in the 4 microseconds roughly that you're spending at each voxel. And so there's enormous amounts of illumination that you require to do that. A spinning disk confocal microscope approaches it differently. It is an array scanner, and you have a micro lens array that basically scans 1,200 points over the sample as it spins. Simultaneously, there's a second set of pinholes through which the fluorescence is imaged onto a 2D detector, like a scientific CMOS or a CCD. And at that point, what it means is that you can collect images, confocal images at 1,200 at 30 frames per second or better, depending on the system you've got. So in addition to being able to go faster, it also is a hell of a lot better in terms of photodynamics. So rather than imaging each voxel, at one time per scan, you're actually imaging it 212 times. You're collecting 212 illuminations, each of which has 1/24,000th the peak illumination. So those of you who thought about fluorescence, and there's probably not many of you that have, one of the problems with fluorescence is that you get saturation. So you end up putting so much laser light in, you get everybody excited, and then all you do is damage beyond that. And so the idea that you can illuminate many, many times at low levels of illumination gives you very high photon efficiency, gives you very high signal to noise, and it minimizes photo damage. And so that's ideal for studies of live cells. Not only is it fast, but it's also very sensitive and very gentle. And so here's a movie, and it's collected at pretty much real time. It's a little bit off. I think it's a little slow. And this is M-Cherry EGFP LC3. These are autophagosomes moving around in this field right here. And the thing I want to point out to you here is that they're moving throughout this entire 2 minutes. They're being imaged at 25 frames per second. And here's the image at 0 seconds, and here's the image at 120. It's just after 3,000 illuminations, the fluorescence is not dimmed, the motility is not changed. These are happy cells at the end of this period of time. And the thing that's nice about it is that you're collecting images fast enough that you can actually collect 3D volumes at something approaching a speed fast enough to characterize things in 3D and motion in 3D. So this is 3D motility of Rab10 containing endosomes in, I don't know what kind of cell this is, probably a CHO cell. It's been a long time. This is a comparison of the kinds of results you can expect from the Yokogawa system, the spinning disk system, versus the Zeiss 510, which is acknowledged and an older system. But after 200 frames of imaging transferrin containing endosomes, there's not much of any change in the fluorescence from the Yokogawa spinning disk, whereas basically you've destroyed the fluorescence from the 510. And these are volumes where time is vertical. And so you can see there's essentially no change in the fluorescence top to bottom here from the Yokogawa system, where basically everything has disappeared within 200 frames with the Zeiss system. So this is a way, way better system for looking at living cells. The system that we've got is equipped for imaging a range of different fluorophores. It's got gas, humidity, and temperature controlled live cell chambers, so it's set up for live cells. It's got an automated stage, which is very cool. I come from a background where we had a dish. We would collect images from that dish, and that was N of 1. The idea that you can identify 10 or more fields and characterize dynamics from that one dish in multiple fields over hours or days means that in one sitting, essentially, you can collect very large numbers of replicates, which is exciting. We also have on-site facilities for cell culture for those of you that want to bring cells to Indianapolis and do some studies. If you're affiliated with the Indiana Diabetes Center, since they kicked in for the upgrade of the system, there's a certain number of free hours available to members of the Indiana Diabetes Center. And I also want to point out that similar systems are found in Notre Dame and IU Bloomington. And my understanding of the other imaging capabilities of the other centers is kind of determined by the CTSI website. So there may be others as well, but that's what I'm aware of. So why wouldn't you want to do spinning disk confocal microscopy? Well, one of them is that it's not very good at all for imaging thick tissues. And that's because when you have all these different pinholes, and this is more complicated than you're going to care about, but all these different pinholes start to interact with each other when you start getting thick tissues. And what ends up happening is you generate a lot more background. So it's not really great for things thicker than cultured cells, essentially. It's also not as flexible. You've got a fixed magnification, unlike a point scanning system where you can use zoom and whatever. And the other thing is you're pretty much stuck imaging a single channel at a time. There are systems whereby you can split up the image into multiple detectors, but at the end of the day, Most people are using this to collect a single image at a time, which profoundly slows the time you can collect multicolor images, because now you've got to change a filter wheel and collect the second image. The second thing I want to talk about is a more complex technology with which I'm less familiar. So we'll see how this all goes. Fluorescence Lifetime Imaging Microscopy. So fluorescence lifetime imaging microscopy is based on another parameter of another dimension of fluorescence, which is fluorescence lifetime. Fluorescence lifetime. And if you look at this Jablonski diagram, you absorb a photon of light, you give a little bit of energy away in vibrational, and then the remaining light is emitted as fluorescence. Well, the period of time between the excitation and the emission is called fluorescence lifetime. And you can measure that through this equation. So the thing that's interesting, and that's not necessarily sounds valuable at all, but fluorescence lifetime is environmentally sensitive. And what that means is you can use that to report various physical chemical properties. And so these are some studies that were done at cuvettes of a probe in different viscosity membranes. And you can see how the fluorescence lifetime has gone from very slow to very, very fast, depending on the viscosity of the environment of the probe. Again, that's not necessarily something that sounds useful to you guys. But the important thing here is that fluorescence lifetime is sensitive to quenching. And so copper quenches fluorescence. And so when copper binds to GFP, it quenches the fluorescence. In essence, what it's doing is it's providing the fluorophore a second path for decay, and the electron a second path for decay, which is basically faster. And so by doing that, you shorten the fluorescence lifetime. That's still not necessarily clear to you why you care about that. Well, you can exploit that for biosensor. So here's an example of the Paradox biosensor, at least version 1, generation 1 Paradox biosensor, which is a sensor of NADH. And in this conformation at low levels of NADH, there's a lot of latitude. The fluorophore has a vibrational energy you can give up through the fact that it's so free in the structure, and you end up getting a longer wavelength. Excuse me, not a longer wavelength. You end up getting a shorter lifetime. There you go. In the presence of NADH, this all firms up. You lose all of that vibrational freedom, and you end up with a much shorter fluorescence lifetime. So this has been used widely to measure NADH in living cells. A second thing that most people use lifetime for with respect to biosensors is for FRET. So fluorescence resonance energy transfer is this process by which you have two fluorophores adjacent to one another, and the acceptor has basically absorbs energy in the range of emissions from the donor. You end up getting this non-radiative transfer between the two. And so for two fluorophores that are in close proximity, you can get FRET. And what that does is you lose the fluorescence from the donor. The energy is translated to the acceptor, which then fluoresces at a longer wavelength. And that's typically how people detect FRET. But the other thing that happens with FRET is by virtue of providing the second pathway for decay of that excited electron, you shorten the lifetime. And so in the presence of FRET, you get a shorter lifetime. And so why would you want to do that? I mean, those of you that have done FRET, I think, can appreciate the fact that it's challenging, but it's not as challenging as going and finding a fluorescence lifetime imaging microscope to do it. Well, the advantage of that is that, first of all, the FRET is reflected in the change of the fluorescence of the donor. You don't need to detect a second probe. So those of you that have done like ratiometric FRET, there's a whole variety of corrections that you have to apply for crosstalk between the two different probes. The fact that all you're doing is measuring the change in the fluorescence of the donor makes it much simpler to do. The second thing is that fluorescence lifetime is a really nice way of providing validation. A friend of mine years ago said that everybody in the world was doing FRET, but probably 70% of the published FRET data were wrong. And that has to do with the fact that it's difficult to detect and it's difficult to validate. And increasingly, people are being called upon to validate it. So if you're doing ratiometric FRET, and here's an example of a paper that we did. This is actually Two-Photon. This is an ACAR pKa sensor. This is the change in ratiometric FRET. And to validate that we were actually measuring FRET, we measured it through a completely separate process, looking at the changes in fluorescence lifetime, which validated it. So increasingly, if you want to do fluorescence resonance energy transfer, to look at interactions between proteins, you may be called upon to do a secondary technique to demonstrate the validity of those measurements. The other thing that's cool about it, and this is going to be where I'm going to falter in my explanation, because this is pi-flim. You can do, because you're just looking at the emissions from your donor, and you don't have to detect the acceptor, you can actually transfect cells with two different biosensors. And to the degree that you can distinguish the fluorescence of the two donors, which isn't that hard to do, you can just use the lifetime changes in those two donors to pretty much simultaneously detect two different biosensors. And the way you do that is by you use a white light laser, and you have a pulse repetition rate of 20 megahertz and every 15 nanoseconds, and you are exciting either one or the other fluorophore. You're simultaneously detecting the fluorescence lifetime in one or the other fluorophore. And by doing that, we were able to distinguish the effects on pKa and calcium in the same cells, which is a real exciting capability. And so this is, I mean, and when I say that you're doing it simultaneously, you're not really simultaneously, they're separated by, 15 nanoseconds, but for all practical purposes, this is a really nice technique for looking at a signaling pathway, if you will. So why not do fluorescence lifetime imaging microscopy? Well, my background was always that the problem with FLIM is that it's slow. It was based on looking at these decay curves. You have to collect a whole bunch of photons over a long period of time in order to generate a decay curve. And then when you're done with that, you're going to have to apply some kind of fitting to this decay curve to estimate a fluorescence lifetime. And frequently that fitting is difficult because the fluorescence lifetime can be complex. So the system that we're using now, and there are a variety of these things, but we're equipped with two of these ISS fast FLIM systems. And I can't explain how this works, so please don't ask me. But basically, it's based on the idea that you're using a modulated excitation, and you're collecting the emissions over time. And so it's called frequency domain fluorescence, and the modulation between the two. You can directly you don't have to accumulate decay curves. You can actually directly measure these from this curve, and you can apply that to something called a Phasor Plot, and that fluorescence. And this right here is just an accumulation of photons that are falling on this point. And this particular point reflects a specific lifetime. So you're not fitting. And the other thing is that basically the quality of the fit has to do with how many photons you've got here. And so you end up generating a spot on that point, but it doesn't have very many points. So your confidence around that value accumulates over time, but you can generate a reasonably confident idea with a very, very short image capture times, which makes it much faster than a gating approach, for example. So this is a really, really nice system. And this is a system that we used. We have two of these things. One of them on a white light laser confocal based system, and the other one is on Two-Photon system. So these are actually measurements of the changes in NADH in the kidney after treating an animal with LPS. And this is endogenous fluorescence, and you can see the change in the range of fluorescence lifetimes between a control and LPS treated animal, which indicated an increase in free NADH under conditions of oxidative stress. The next thing that I am much more conversant on is to do with intravital microscopy. So why intravital microscopy is microscopy of living animals. Coming from a background in cultured cells, I was horrified by the idea of having to work with fuzzy little creatures, but why would you want to do intravital microscopy? Well, those of you that study cultured cells, I think you appreciate the fact that what you study may or may not have much relevance to an animal, to be honest, much less a human animal. I spent years studying MDCK cells, which are these maiden Darby canine kidney cells that came out of a Cocker Spaniel in the late 50s. And from that, I'm trying to extrapolate to what human physiology is about. And so it was a kind of a leap of faith that everybody that studied it kind of agreed to make and not question each other over. But the idea that you can look at cell biology in vivo in living animals is very exciting. So first of all, it's more relevant. I think you don't have to make any apologies for it. The other thing, it means you can look at cell biology in the context of a disease, which is very difficult to reproduce in cultures. You can look at injury models, and you can look at immunology. And all of those things are very good justifications for intravital microscopy. You can also look at the cell biology of cells within an intact organ. You can look at interactions between cells and tissues within an organ. And for that matter, you can look at organ function. And then you can look at studies of dynamics. So if you want to look at things like cell motility or blood flow, those are things that can only be evaluated using intravital microscopy. And then finally, and a lesser appreciated thing is that you can imagine doing, for those of you that do studies of immunofluorescence of fixed tissues, there are plenty of molecules that just don't stay put during fixation. So the idea of being able to look at the distribution of proteins or of targets in the living animal is very exciting for things like siRNAs, for example. So how do we do intravital microscopy at the center? We use something called two-photon microscopy. And the thing about two-photon microscopy is that whereas confocal is a cuvette with something fluorescent in it, and on the left you're exciting that cuvette with visible light, and you're stimulating fluorescence, and this is basically the cone of light you see over here. You're stimulating fluorescence everywhere in that whole range, so it's being focused down to a point, and then the light proceeds onto the other side of the cuvette. You get fluorescence everywhere. Because of that, you need to have a confocal pinhole to reject all that out of focus fluorescence. In contrast, in two-photon microscopy uses infrared light, and this is more detail than you care about probably, but the point is the infrared light doesn't have sufficient energy to excite fluorescence by a one-photon process. Two photons have to arrive simultaneously in that sub-femtoliter volume in order to stimulate fluorescence by this two-photon event. But the beauty of that is because that only occurs at the focus. It means you're not stimulating any fluorescence above or below that one final point. So now all of a sudden, first of all, IR light penetrates into tissues better, but the other thing is you don't need a confocal pinhole to reject all that out-of-focus fluorescence. And for that matter, you know exactly where the fluorescence is coming from. So you can surround the sample with all kinds of detectors and collect all the light that's scattered by the tissue. So for imaging into biological tissues, which are scattering, the idea that you can collect that scattered light and make an image out makes two-photon microscopy the method of choice for intravital microscopy. This is an example of, so I don't know, it was like 2002, I believe, we first started doing two-photon microscopy. This is an image collected from the kidney of a living rat. And what you see over here is several probes. Herx is labeling the nuclei. This is a glomerulus. So let me see. There's a kidney, there's a nephron, there's a glomerulus. And so what you have here basically is an optical section from a live animal going through glomerulus and some of these tubules. And what you can see here, here's the capillaries and the glomerular tubule. This is the first segment of the proximal tubule. So you see endosomes where the proximal tubule has taken them up. And these are subsequent segments that have also taken up a green dextran that's been filtered. And then it's been accumulated in this distal tubule because the business of the kidney is to reclaim a lot of things. One of the things it reclaims is water. So when the probes go down here, water gets reclaimed, and what comes back up and appears here is very bright. This is an example of a video that we collected years ago that was just astonishing to most nephrologists, because this is basically demonstrating a process that nephrologists inferred occurred, but they actually never saw it, and that is glomerular filtration. So in the beginning of the movie, we inject the animal with a combination of a green dextran that's too big to get filtered out of the glomerulus and a red dextran that does get filtered. And you can see the rapid filtration. And we've parlayed that into a lot of different studies since then. And again, this is the foundation of this O'Brien Center that we've had funded since 2002, whose goal is to develop and disseminate techniques of intravital and 3D microscopy. We also then parlayed that into the ability to image the bone marrow of the mouse Calvarium. And here's an image collected from a living mouse. This is a mosaic of the Calvarium. And this is a movie, 3D volume showing blue is second harmonic from collagen on the surface of the bone. Green are the hematopoietic cells, and red is a dextran that's flowing around in the vasculature. It's been funded in 2007, and again, to develop techniques of intravital microscopy of the bone marrow microenvironment. This is more recently, we were funded to be a core of the NIH Indiana Diabetes Research Center to do imaging in support of studies of diabetes. This is an example of an image collected from a knockout mouse. This is pancreas. This is an image collected from islets transplanted into the mouse kidney. This is a technique that's being used by one of the members of the Senate to look at human islets. So there's a resource for human islets that can be transplanted into mice and behavior, essentially doing intravital microscopy of human islets, if you will, transplanted into the mouse kidney. And then more recently, we've been working on the effect of diet on signaling in muscle, and this is a phosphatidyl acid sensor that detects changes in phospholipase D. And you can see these muscles in the anterior tibialis muscle. My lab does a lot of intravital microscopy of the mouse liver. These are NKT cells, I believe, migrating around inside a liver 24 hours after introduction of LPS into the animal. So it's a septic animal, if you will. This is a study that we did with Lilly, looking at this disposition of bifunctional antibodies, which are taken up into endothelial cells. It's a study that I did for Amgen, looking at the effects of various drugs associated with idiosyncratic drug-induced liver injury on hepatic transport. And this is a study that we did with the Biocomplexity Institute in Bloomington, looking at the effect of acetaminophen, which is known to kill your liver. But the interesting thing is we looked at the effect on mitochondrial membrane potential. Mitochondrial membrane potential sensors accumulate on the base of function. So this region, these are dead cells, they're accumulating propydium iodide. And the region around these cells, there is this region of lost membrane potential. But within like 30 minutes, they recovered. And so the dynamics of this was something you're only going to be able to see using intravital microscopy. So why not do intravital microscopy? Well, first of all, it's expensive. I mean, the instrument itself costs something on the order of $800,000. It's technically challenging. You need people that are willing to deal with developing the techniques of not only the microscopy, but the surgery and the maintenance of the animal and all this other stuff. So why would you, I think we've addressed a lot of those issues. We have the wherewithal to support surgery, the microscopy, the image analysis, and provide support for IACUG protocols, which are obviously an important thing. We've also got an enormous amount of experience doing intravital microscopy. And I'm not going to go through all these different things, but kidney, pancreas, liver, mouse calvarium, muscle, mouse skin, lymph node, in collaboration with all these different investigators. So we've got lots and lots and lots of experience. We've got 3 multi-photon microscopes, including a new one that we got just a few years ago now from Leica. This is the latest, greatest system. And again, this is the thing that I used a moment ago to show you that each one of these green spots here is a mitochondrion, an individual mitochondrion in hepatocytes. And these red things here are endosomes, where this IgG has been internalized. The thing that's cool about it is it has these special spectral external detectors, which allows us to tune the emissions. It's high-D detectors, which are a more sensitive, more time-based detector that's very powerful. There's 2 IR lasers. The 2 IR lasers are useful because we've been, up till now, restricted to imaging things that can be excited up to something on the order of 1,000 nanometers by a titanium sapphire laser. This new laser brings us out into a range of wavelengths that you can see. You can start imaging red fluorescent proteins. It also allows us to do dual excitation. So this is the simultaneous excitation of tetramethylrhodamdextran and that's not true. It's tetramethylrhodamine labeled islet and actually fluor-647 albumin in the vasculature. So you can look at 2 different probes that are excited in different ways. And the other thing is, it's pretty commonly known that the longer wavelengths penetrate deeper into tissue. So this is an image of the kidney, I believe. I'm not really sure what I'm looking at here. And imaged 800 nanometers versus 1200 nanometers, and you can image much more deeply at longer wavelengths. So we're really excited about this new system. The other thing is through the various cores, we've developed a whole range of biosensors and ways of introducing them into living animals. So we can assay things like free calcium, cytosolic calcium, reactive oxygen, protein kinase A, AKT, phospholipase D. We've got FRET standards as well, these constructs you can use to actually validate your FRET measurements. And then we've got a variety of different fluorescent fusion proteins, including GFP and RFP GLUT4, LC3, like I was telling you a moment ago. Here's some examples of some studies. This is glucagon-mediated protein kinase A activation in mouse liver using the ACAR biosensor. These are synchronized calcium transients as measured with GCAMP6S. This right here is, what is this right here? This is Rho GFP2, it's a reactive oxygen sensor. And Amelia Linneman's looking at this, and she's seeing that there's a specific reaction in a subpopulation of cells within the islet that she's interested in. And then the other thing, the last thing, those of you that are familiar with intravital microscopy probably think in terms of the other advantage that you can do longitudinal imaging. And that's something that people publish one paper on and then they're done. And that's because it's a challenging thing to do, to put a window into an animal that allow you to come back and look at that animal over a course of weeks. It is something that we've accomplished. We actually sent Gosha Kamoshka over to Europe to learn how to do this. And you can put a window in the animal, and we've imaged the pancreas over a period of 32 days. We've imaged the liver after 14 days and the kidney after 21 days. This is an image of kidney. The white is second harmonic imaging of collagen. The green is GFP lyse-expressing immune cells, and the red is a dextran of the vasculature. So we have a lot of the things that you would need to actually put intravital microscopy in play. That would be otherwise be deal breakers for most people trying to do it. So we're unusual in the sense that most people that do intravital microscopy have a lab devoted to it. So the idea of having a core resource to support this and to support it to the degree we do makes it, I think, really unusual. Tissue cytometry. Tissue cytometry is, I think, an exciting new thing that we've gotten into recently. And we're now funded by, I think, 3 different core grants to do this. Tissue cytometry is basically what you're doing is you're taking very, very large images and identifying individual cells within that tissue, and then characterizing the amount of fluorescence associated with each one in multiple channels to identify the different kinds of cells. And then you're doing just the same kind of thing as you do with flow cytometry, except you have spatial localization of these things. So this is an image of mouse kidney that was collected by Tarek Ashkar's lab and was rendered in Imaris. And the thing that's cool about this is that when you combine this with techniques of tissue clearing, and tissue clearing is this technique that was actually developed at the turn of the 20th century, where you remove the refractive index disparities in tissue to generate images, to facilitate microscopy of complete 3D volumes in the scale of millimeters. When you combine tissue cytometry, the ability to collect these large scale images, with tissue clearing, which allows you access to millimeters of depth, then you can suddenly do a complete census of all the cells in an organ. I should have started this movie before. This is not our work, by the way, but it's just so astonishingly beautiful. I wanted to share it with you. This is an image of a lymph node that was collected by Michael Gerner, who's now in Seattle, showing the distribution of different fluorescent probes, different cell types. And when you combine that with various techniques of advanced image analysis and data analysis, you can start doing things like neighborhood analyses, and you basically generate the same kind of scatter plots that you would by flow cytometry, but it has the additional benefit of giving you these neighborhoods so you can better understand what these cells are actually doing. So rather than, for example, you take a representative field, which tells you something, you're actually collecting a complete census of all cells in an organ of a mouse. And then it means you have the ability to detect and characterize rare events. You can look at interactions between different structures and different kinds of cells at different scales. So there are structures that are just not captured except in 3D. And then when you, like I said, when you apply advanced methods of data analysis, you can actually go beyond the questions that you go into the problem with. You can start identifying new patterns that you hadn't even thought of when you started the experiment. So it's very, very exciting technology. We're doing that as part of this very cool NIDDK precision medicine project, where we're getting human kidney biopsies. We and a bunch of other labs are getting kidney biopsies from people with kidney disease and applying all the latest techniques, in our case, multiplex fluorescence microscopy, proteomics, and spatial transcriptomics, to characterize really fully these kidney tissues from these people with kidney disease. We are now also doing Codex for this, the Center of Excellence in Molecular Hematology. As another center, this is some tissue cytometry of the mouse bone marrow. And an important component of this is not enough to collect the data, but you have to be able to analyze it. And so we have various kinds of software. There's Koia MAV software, and I'll tell you about a Koia in a minute. Indica Halo is very, very expensive image analysis software, but it does very, very, very high level analyses, image processing, segmentation, and artificial intelligence-based cellular classification. It's pretty much cutting edge technique. And some software that we developed ourselves. This is based on ImageJ. This is freely available to anybody who wants to do it, that allows you to actually interactively look at your images, process your images, do supervised analysis using gating techniques like you would flow cytometry, looking at the distribution of kinds of cells, or alternatively doing unsupervised analysis. So you can do a T-SNE analysis to identify different kinds of cells and map them into the tissue, and then we'll go back and forth. So this is very, very powerful. Again, it's available in ImageJ, so anybody can use it. Anybody has access to it. Another technology I want to talk about today, I think I'm doing okay on time, is Codex high multiplex fluorescence microscopy. So why would you want to do something called Codex imaging? All right, so the volume I showed you over here earlier is this work that we've done with multispectral imaging of kidney biopsy samples. We're using linear unmixing. And you need to do, and the reason for that is that fluorophores, once you get beyond 2 or 3 probes, the fluorophores emission spectra overlap with one another. So it's very difficult to distinguish one fluorophore from another. And it reaches a point where you have enough probes that you can't distinguish one from another. And that's the upper limit of what you can do with conventional fluorescence microscopy. You can do linear unmixing, which gets you to the point where you actually can deconvolve the composite spectrum that you get out of these kinds of images to identify specific contributions from specific probes. But at the end of the day, it's very difficult. It's highly incredible when you do more than maybe 5 or 6 probes. So the alternative approach to this is one where you don't label the tissue with all the different probes at once, but which you actually do sequential imaging. And so there's a technique called Cyclic Immunofluorescence, where you label the tissue with 3 probes. You bleach the hell out of those probes. You label them with another 3 probes. And you collect another set of images. You bleach the hell out of them. And you keep on doing that. And you add up an image that has multiple rounds of fluorescence collected and multiple probes. And you can do 20 or 30 probes that way. The technology that we brought forward is that we've got on the core is called Codex. We got money from the Department of Medicine and a CTSI core equipment grant to buy the components of this. Essentially, you've got a high throughput microscope over here, and here you have a robotic image liquid handling system over here. And the way it works is very slick. First of all, it's based on antibody cDNA oligoconjugates. So you've got those in hand. You incubate your tissues with 20 or more of these things. You add DAPI, and then you add 3 cognate Codex fluorescent oligonucleotide probes. So in other words, there's 3 different colors. There's green, red, and far red. And each one is attached to a specific sequence that will recognize one of those 20 antibody cDNA oligos in the tissue. You throw those on, they bind to their cognate probes. You rinse all the probes off. You collect a 4-channel image of the DAPI and those 3 probes. You strip them off, and you do it again with the next 3 Codex reporters. And you do that over and over again. And you can image 30 or more probes. And this was developed by Gary Nolan at Stanford. And this is a movie that is really difficult to get to play. But what is happening in this movie, hopefully, is you're seeing the same field. This is 3 replicates of 3 different conditions. And what we're doing is we're scrolling through different pairs of probes. Do that again. Or try to do that again. Perhaps not. Anyway, at the end of the day, you can generate an image of, this is a panel of 56 antibodies. So he generated 56 of these oligonucleotide probes. And this is an image of the same tissue for all 56 of those different probes. So he conducted, this is pretty heroic, he conducted Codex imaging of 140 samples from 35 advanced stage cancer patients, colorectal cancer patients. And then he identified from these images 9 conserved distinct cellular neighborhoods. So again, this is a matter of applying some advanced imaging and data analysis techniques to identify cellular neighborhoods, specific kinds of cellular neighborhoods. And the thing that was neat about it is that they identified a cellular neighborhood that could identify an increased level of survival in a high-risk patient subset. Now, from the perspective of the patient, this is very neat because what this means is that you just gotten bad news, that you are one of the people you're in this high-risk patient. So you're assuming that you've got a very limited lifespan. This is one more piece of information that could be applied to that to tell you that either you are or you are not in a high-risk patient subset, that this is something can be used to distinguish in stage treatment and various things. So it's very cool because it pulled out additional information beyond that which could have been obtained by standard techniques. It also discovered that there were inferior outcomes that are associated with coupling of tumor and immune cell neighborhoods. So again, the analysis is this very heroic, large-scale analysis that Nolan did, identified factors that are going to be important in terms of treatment and staging of patients, if you will. So it's a very exciting technique. It's also a very complicated technique. We have, nonetheless, like I said, we've acquired a system here. This is an example of Codex imaging of kidney that we're doing as part of the O'Brien Center, as part of this NIDDK Precision Medicine Project. And this is an example then of 23-plex Codex imaging of the human kidney. We've just now applied for renewal, and this is something we're going to be doing nationally, both for this KPMP project. In terms of implementing Codex for IU researchers, this is something that you're not going to want to do on your own. First of all, the system is reasonably expensive, but it's also something that the reagents that you get require a lot of understanding. We need a lot of understanding of image collection. There's a lot of expertise in image analysis required for it. And to be honest, the reagents that we got from Koia are very variable in their quality. So we've got a lot of experience with all of these things. And so we can facilitate, not only do we have the system here, but we have this backlog of expertise and experience that helps us use this. And the other thing is that it's not just us. There are a bunch of labs on campus. So right now there are, I don't know how many this is, 1, 2, 3, 4, 5, 6, 7, 8. It's 5 labs that have actually done 5 or more runs each. Is the number of targets. I think there's something like 12 different labs that are currently pursuing Codex studies, all of them with a certain amount of experience that we can use. A lot of them using a lot of the same probes. And the other thing is there's a national Codex work group at Vanderbilt. Diane Saunders put this thing together. She does diabetes research. And they've got this wonderful resource of antibody clone lists that they've validated what their experiences were. And also they've generated so many of their own antibodies and they've validated those. So in other words, when you want to get into this, it's really great to be able to interact with her and say, what antibodies you used to do this, what worked, what didn't work, and more importantly, what didn't work, because you don't necessarily find that out. So we have a lot of the infrastructure in place to enable people to do Codex image. Why wouldn't you want to do Codex imaging? One of them is the cost. If you're going to be doing 24 probes, just by virtue of the fact that you've got antibodies, 24 antibodies is an expensive thing. You're going to be pretty heavily invested in the whole thing to get involved with it. The Codex conjugation of antibodies makes them even more expensive. We're estimating each antibody costing on the order of $700 to $16,000 just to put the probes together. Then it costs some money to do the run. And so to do 1 2 centimeter by 2 centimeter area in 24 probes costs $19,000. To do 3 replicates, you're talking $23,000 because you've already bought the antibodies and then you have the additional cost. So it's a very expensive proposition. Like I said, it's not something that it scales with probe number because there's a fixed number of cost per imaging run. And it doesn't scale with replicates because there's the one-time antibody cost. And it also assumes that you're doing 1 sample per run. So to the degree you can fill up your 2 centimeter by 2 centimeter area with more tissue, then the costs obviously go down. That said, one of the things we want to do is, although we've got a lot of people interested in doing Codex and actually actively doing it with us, we would like to open it up to more people. There's a group of people that have identified a need, but have kind of gasped at the cost. And so what we want to do is, and Koia just recently capriciously increased their pricing. And if you don't like it, then don't do Codex, is Koia's attitude. We were trying to address the paralyzing cost of entry. I think once you've incorporated Codex into your imaging and into your research, then that means that you can apply for grants and fold that into your grant proposal. But how do you get the data to begin with? Well, the CTSI is one source of funding, and there's been 2 projects funded by the CTSI to do this. But the other thing we've done is gotten money from the CTSI, from the university, to basically develop a test kit for pilot studies. We can provide people with free antibodies to try out in their tissues. And then we've also validated homemade reagents, so we don't have to buy these expensive reagents from a Koia anymore, and we're providing those recipes to different labs. The other thing is that a challenge to Codex is that it's been enormously popular and it's enormously time consuming. The KPMP just funded us to do 40 biopsies per year. We just now got funding for this HubMap project for looking at normal kidneys. We're also providing this as a service for the O'Brien, the Center of Excellence Hematology, and the Diabetes Center. So all these things combined, there's going to be a lot more use of this. And so today, actually, Connor Gilbronson's up in Chicago seeing a new system that's supposed to have 5 times the throughput at much less cost. So we're hoping we're hoping their promises are actually true. So future plans. This is turning out perfectly. Super resolution microscopy. Why would you want to do super resolution microscopy? I've actually reviewed a lot of grants for the NIH on shares mutation grants where people propose they want to do super resolution microscopy. And they want to do super resolution microscopy because it's got more resolution. Obviously, more resolution is a good thing. But by point of fact, there are a lot of things that you don't need that additional resolution for. And so you have to ask your question very carefully. Really, are you seeing anything more? And I would maintain that a lot of the images you see of the results coming from super resolution are just sharper images of things you could see anyway. So there's a very restricted number of applications, I think. So 200 nanometers, for example, is about the best resolution you're going to get from non-super resolution techniques. And that's not enough to look at the structure of mitochondria. It's not enough to resolve the structure of endosomes. It's not really sufficient to resolve interactions at neural synapses, nuclear pore complexes, glomerular slit diaphragms, for those of you that are nephrologists. Electron microscopy is great. You can get enormous resolution out of it, but that's typically for a single probe. And people will argue with me about that, but it's challenging. You're only going to do it in fixed tissues, and 3D is something that is a heroic epic thing that you can do, but you're not going to do a lot of it. So super resolution resolves anywhere from 20 to 150 nanometers, but it does that with compromises. The compromises include speed, photo damage, multiplexing, simplicity, and reach, how far into a tissue you can look. So the question is, when you're looking at super resolution, what are you willing to give up for this additional resolution that you think you need? So one method of super resolution involves single molecule imaging, where basically what you're doing is you have probes that are emitting at a low enough rate that when you collect a fluorescence event, you know it's coming from a single molecule. If you collect a bunch of images, add them all together, and you know they all come from the same point, then at that point you're safe in saying the middle of this distribution is the place the fluorescence is coming from, and you can resolve it to whatever you can get from the number of events you've got in that Gaussian profile. It has very, very high resolution. You get 20 nanometers lateral and 60 nanometer axial resolution. It's relatively inexpensive. It's not very complicated. It comes down to the probes. Really, what you're doing is collecting a whole bunch of images and then adding them up with one another. The downsides is that it's difficult to do multiplexing. The reach typically is restricted to 100, 200 nanometers because you're normally doing total internal reflection fluorescence to excite one focal plane. It's slow because this whole idea is you're basing it on accumulating a bunch of images. So you need to collect 100 is ridiculously low, but it's more like 1,000 to 10,000 images. So you're not going to do it in live cells. You're also not going to be able to do it even in fixed cells unless your stage is incredibly stable. The live cell studies are also compromised by the fact that it's difficult. A lot of fluorescent proteins don't behave optically or photodynamically the way that they need to be compatible with this technique. And in the end of the day, the success depends on a lot of significant optimization. So in putting together talks that I've done in the past, I found that a lot of the images that I used for my talks came from one lab. And I think that reflects the fact that it's not very damn easy to do. It's something that people do it. They are dedicated to it, and they develop the technology and they use it. So it's something that you would think would be applicable in a core resource. But my interactions with people at different core directors is that they've acquired these systems that have since become doorstops because very few labs are willing to invest the time into the probe chemistry that they have to do in order to actually make this work for them. Another approach that's much simpler is structured illumination microscopy. Oh, and in terms of Purdue and IBRI both have systems. The Indiana Biological Research Institute both have STORM systems. I believe both of them. Purdue's, I think, is a Nikon system. The IBRI is a Zeiss system. Structured illumination microscopy is compatible with any probe. It's easily extended to multiple probes. Imaging is pretty much turnkey. It involves looking at the interaction. You collect different images. You make an interference pattern between these different images, image at different orientations. And by that, you interpret the interference pattern in terms of a higher resolution image than any one of those images would generate for you. The problem with it is that the increase in resolution is incremental. You're going from 200 nanometers to 100 nanometers lateral. It's somewhat limited reach. You can't go very deep into the tissues, no more than 10 microns. It's also slow because you got to collect a bunch of different images for each color, which makes it incompatible with rapid dynamics. And there's been improvements. So the resolution has been improved, nonlinear excitation, higher speed with spinning disk or instant sim. I don't believe this is commercially available. And you can also get more reach and speed through lattice light sheet microscopy, which is something that up until recently has been a research technique rather than a commercial technique. But my understanding is that Zeiss has now implemented this into something that's actually usable by regular human beings beyond Nobel Prize laureates. And finally, what I want to spend most of my time is talking about stimulated emission depletion microscopy, which is a point source engineering technique where you basically shave down the excitation. And I'm running out of time, so I can't really talk about that very much. At very high resolution. Images can be collected rapidly because you're essentially just doing confocal microscopy with a small focus, if you will. It's compatible with imaging, the studies of live cells, has fewer stability issues, essentially turnkey, and it's 3D to a point. Cons are you limited to colors. It involves an enormous level of illumination. And the first commercial versions of the system were just not really very useful at all. And that had to do with the fact that by virtue of the way that it works, you're actually doing a lot of damage to the fluorescence and to the tissue. Newer systems from Leica and Bier both have various things they've improved through using lifetime to augment the resolution and decrease the illumination. And the other thing has been challenging is limited reach. Any kind of optical aberrations, which is completely screw up the point spread function. Leica has shown some really nice results, and Bier as well, in clear tissue and using optical, adaptive optics. So we got a high-end instrumentation. I submitted a high-end instrumentation grant last year. My understanding is that it's probably funded, and it's going to be coming through in fall of 2022. So we should have that system. The last thing I want to talk about in the no time I have left is large-scale imaging by light sheet microscopy. All right, so tissue clearing. I told you about tissue clearing. It was first described in the 20th century. It's involved removing refractive index gradients. And by doing that, you end up you can collect images much more deeply because there's no more scattering happening in the tissues. Now, my interest in light sheet microscopy has more to do with the fact that confocal microscopy is ridiculously slow. This image that I showed you earlier, the 16 channel, 4 millimeter by 11 millimeter by 50 micron volume collect required 36 hours of confocal microscopy to collect this image. An alternative, and the bottom line is that confocal microscopy is just a really silly way of doing large scale microscopy. You're imaging at 1 to 5 frames per second. Volumes can take days to acquire. And if you're doing multiple replicates of multiple conditions, you might be taking months just to collect the images, if you will. So it's just not very feasible. So a lot of the really large scale images you've seen were either collected using light sheet microscopy, or alternatively, there are labs devoted to this, like Gerner's lab. Light sheet microscopy. So rather than using a pinhole to reject out of focus fluorescence, you actually illuminate only a single focal plane. And that focal plane is projected to the sample orthogonal to your image, and you collect an image of that single fluorescence from that single focal plane onto a 2D detector. Now that you're detecting on a scientific CMOS, you can collect images at about 100 frames per second. Not only that, but because of the fact that if you're imaging at 100 frames per second, that gives you integration times that can be like, rather than integrating for 5 microseconds per voxel, now you're integrating for 50 milliseconds. That's 10,000 times more dynamic range. So it makes it very gentle, very fast. It's great for live dynamics 3D. And that's basically what it was described for initially, was the idea that let's use this for imaging tissue over time. It's very gentle, very efficient. And so this is Drosophila development over a period of 2 to 5 hours. This is Zebrafish development, and I wish I remembered what we're watching develop. I believe these are neurons extending out over time in a zebrafish embryo. So it's great for long-term imaging of tissues, but it's also really great for imaging high-speed events. This is a beating zebrafish heart. It's collecting images so rapidly that you can actually characterize this in 3D. So it's a terrific technique for live tissues. But the point I want to make is that it's also fast enough to make large-scale, high-resolution microscopy practical. So whereas I was telling you that it would take months to do a full study by confocal microscopy, you can collect a 1 millimeter cube at 1 micron resolution in 9 hours using a confocal microscope, or in something between 1 and 18 minutes by a light sheet microscope. And the time to image that volume I showed you before goes down from 40 hours to 2 hours. The problem with light sheet microscopy is that, and I don't want to highlight the problems with respect to these other systems, because this system is actually working at Notre Dame, and this system is present at the VA at IU, is that you have to come up with a way to mount your sample. And typically, you have a sample, you're projecting light from the side. The sample has to be somehow mounted high enough up that you can project a light sheet through it. So it's not really compatible with slides, although Leica has tried to make it possible. So you have to come up with these various techniques. You're either impaling the tissue on some kind of rod, or you're putting it in agarose and spinning it around, or you're mounting it in some kind of horrible clamping device in order to present the tissue for the labeling. And again, I'm not dismissing that, because that's something that can be worked out for various tissues. But for the point of doing imaging of slide-mounted tissues, John Liu came up with an alternative approach, which involves basically rotating the whole thing on it by 45 degrees and putting everything below the focal plane. So now you're projecting the light sheet through the sample at one angle, and you're collecting the image orthogonal to that. And this is a video that his lab put together of the system that he built to do that. And I'm only going to let you go through the first part of the video because we don't have time. And how simple it is to mount the sample. You put your slide in, you go in and you grab your tissue, you drop the tissue, you drop some more tissue, and essentially you're off to the races, ready to do light sheet microscopy. So it's a very, very nice, efficient system for doing light sheet microscopy. And here's a sample being scanned at high throughput. This is a 3D volume collected in an hour at 4 micron resolution of an entire mouse brain using this system. This is cleared in ethylcinamide. And I would let this whole thing go, but we don't have time. Another thing is very cool that John Liu is pushing this for is utility for pathology. And so those of you that have had any experience with prostate cancer, like me, would appreciate the fact that you really, really want to know the diagnosis is correct before you make a decision. So in looking at single sections of a given prostate sample, 2 pathologists had an inter-observer error of 47.5%. It was like a toss of a coin as to whether or not it was stage this or stage that. And for a person that's either going to get their prostate removed or not, that's a really critical thing to know. And so what John is promoting here is the idea of using 3D cytometry, if you will. This is light sheet microscopy of this prostate sample. There's your benign glands, apparently. And somewhere in here, I believe there are going to be some cancerous cells, I hope, soon. Anyway, after evaluating this entire 3D volume, the pathologist agreed completely on a particular score. So the point to be made here is that the 3D visualization of this entire thing, there's the adjacent carcinoma cells. They could not have possibly agreed on this, but when they applied this technique and got data from this 3D system, they were able to distinguish this and actually make a good call on for diagnosis. So the system that we're talking about getting, and I'm sorry that I'm going over my time here, involves 2 different light sheets. And the idea of it is that you're going to combine a low resolution fast screening with a high resolution cellular characterization of let me see if I can't get to that point in the video. Yeah. So there's two different light paths. One providing large scale, very quick illumination of a sample. So you can collect this entire 3D volume very, very rapidly. And then later you come back and you collect a second image at much higher resolution. And the thing that was cool about this is that he used that, the way that he demonstrated the utility of that was in a sample with metastatic lesions. Let's see if I can't pull this up. There we go, right? So here is an entire mouse brain imaged by light sheet microscopy. And there's a variety of cancer lesions. And again, you wouldn't necessarily find all these in any 2D section. But the challenge is that imaging these at high resolution would still take like 2 weeks. So the idea of collecting a low resolution image from which you can identify regions of interest to characterize a higher resolution has reduced the time from confocal microscopy, 228 days, down to 2 weeks by light sheet microscopy, down to 5 hours by this hybrid to light sheet microscopy. So that's all very fine. We don't have one, but we just put an application in April 2022 in response to an RFA for Project to Image the Joint Innervation. In 2022, we're going to be renewing our O'Brien Center. We're going to be looking to get one there as well. So we're likely, I think, to have one of these things in our system soon. Using the ICBM, we charge $60 an hour for our major microscopes, $25 an hour for image analysis systems. We have special rates for long-term studies going over the weekend. The idea is to contact us, and I'll show you how to do that in a second. We can help you identify the most appropriate system or an approach, or whether you shouldn't do microscopy at all. We provide training in the appropriate microscope system, and at that point, you're either able to use the system absolutely independently, or if you can get as much help as you want, all the way down to someone actually doing studies for you at the rate of $60 an hour. There are various sources of money. CTSI has money twice yearly to support pilot funding of research use of core facilities. We've got lots of money this way. A lot of our investigators have. You get $10,000 over 2 years, which is plenty of funds to get preliminary data. There's also something called the CTSI Postdoc Challenge. It's $5,000 for 1 year. The application deadline is called September. The CTSI website is not current. I'd like to put a bug in Jill's ear about that. And there's CTSI project development teams, which are really, really valuable for identifying what kinds of things you need to do to make your research projects better, your proposals better. You can get something up to $10,000, $15,000. There are also two centers. Our O'Brien Center has fellowships. They can get something up to $10,000 to do studies. And the Diabetes Center has a pilot feasibility program whose deadline just recently passed. We just missed the boat on that. To do something with us, this is our website. Hopefully it'll be updated soon because it's way out of date. You would contact Gosha Kamoshka, Connor Gilbronson, or myself. And with that, I will end 8 minutes late. Does anybody have any questions?
All right, thank you, Ken. That was a really great seminar, and thanks for highlighting all the CTSI funding mechanisms.